Hi friends, my name is Fanindra. So in this video, I am going to explain about Plasmodium vivax in man phase. I already done a video on this topic before 3 years. And again, I am going to do this now because I have explored a lot of information regarding this topic and I am going to share it with you people right now. So a lot of information I'm going to provide it for you. So watch this video till the end. You can clearly understand about Plasmodium vivax in man phase. So generally this Plasmodium vivax will occur in two phases, that is in man phase and mosquito phase. I mean it occurs in the human being, either it may be a female or male. It occurs in the human being, and in another phase it occurs in mosquito. So basically this Plasmodium is a parasite which causes a disease called as malaria. Okay, so now listen properly. So basically this Plasmodium undergoes the process of asexual reproduction and leads to the formation of zygogony. Okay, so normally this Plasmodium will infect liver cells and it also infect the red blood cells in order to cause a disease called as malaria. So once they enter into liver cells, I mean we also know that these liver cells are also called as hepatic cells, which are mainly present in the liver, right? So hepatic cells or liver cells, both are same. So now once this Plasmodium enters into liver cells and start destroying them, then it is called as hepatic zygogony. As it is occurring in the hepatic cells, it is called as hepatic zygogony. And once it entered into the RBC cells and started affecting this RBC, then it is called as erythrocytic zygogony. Right? We also know that these RBC cells are also called as erythrocytes. Hence, this Plasmodium which affects these RBC cells, then that process is called as erythrocytic zygogony. Right? Till here, I think you are cleared. And basically the process of this Plasmodium vivax in man phase was discovered by Schott and Granham. Okay, so now here how this Plasmodium vivax in man phase begins. See, generally we know that we are humans and mosquitoes will bite us. They will suck the blood from us and they will also release the waste material which is present in it. Okay, so now what this mosquito will do? That mosquito, let us say that mosquito is Plasmodium infected, which means the Plasmodium parasite is present in the gut of that mosquito. Hence, we are going to consider it as Plasmodium infected mosquito, right? So now once this Plasmodium infected mosquito, which consists of Plasmodium, will go and bite human being, either it may be a male or else either it may be a female. It doesn't care. So this Plasmodium infected mosquito will go and bite this human being. Then what will happen? Then it starts releasing 2000 sporozoites. It starts releasing 2000 sporozoites. That's nothing but the Plasmodium itself. Okay. So the 2000 sporozoites will enter into the human being. So where does it enter? It enter into hepatic cells and RBC cells, which I have said you before itself. Right? So once it enters into the hepatic cells, it causes hepatic zygogony, and once it enters into the RBC cells, it causes erythrocytic zygogony. Right? So now this Plasmodium infected mosquito will bite the human being in order to release 2000 sporozoites. And now here the actual story begins. You know what are the stages which happens in this human being? See, pre-erythrocytic cycle, exoerythrocytic cycle, pre-patent period, erythrocytic cycle, incubation period, formation of gametocytes. So these are the stages which are involved in the Plasmodium vivax in man phase. So now we have to listen properly. I'm going to explain the life cycle of Plasmodium vivax in man phase. Listen properly. So now let us learn about the life cycle of Plasmodium vivax in man phase. As I have previously said you, this Plasmodium infected mosquito, once it bites human being, then 2000 sporozoites will be released. So now once mosquito bites it, then 2000 sporozoites has been released out, right? And now that released sporozoites will enter into the hepatic cells. It will enter into the hepatic cells. So all of these are nothing but the hepatic cells. Okay, and these are nothing but the red blood cells. Okay, so these are hepatic cells. So now once these porozoites which are released by the mosquito will enter into the hepatic cell, then what will happen? Then it will get transformed into trophozoite. It will get transformed into trophozoite, and now this trophozoite will consume the content which is mainly present in the hepatic cell, and it will get developed into a schizont. And this schizont is nothing but which is spherical in shape. Okay, and it is completely developed. It the size of that schizont will be increased when compared to that of trophozoite by consuming the content which is mainly present in the hepatic cells or liver cells. Okay, so now the schizont will undergo the process of mitotic division, right? It undergoes the process of mitotic division, and even the cytoplasm will undergo the process of the division, mitotic division. I mean, which is also called as cytoplasmic division, in other words to say. Okay, so that mitotic division will be done in such a way that the nucleus as well as the cytoplasm of the schizont will get divided several times in such a way that cryptozoites will be released. Okay, and now how many cryptozoites? 1200 cryptozoites, right? And what is the other name of these cryptozoites? First generation merozoites. They are called as first generation merozoites. And now this process till now, what has happened, and that process is called as pre-erythrocytic cycle. Okay, and what is the time duration of this pre-erythrocytic cycle? 8 days. Okay, how many days? 8 days. And now these cryptozoites will have two options. So what are those options? See, this can enter into the fresh hepatic cells. And continue the exoerythrocytic cycle. This is one of the option. And another option is that they can enter directly into the RB cell. I mean this red blood cell. Okay, they can directly enter into this RBC cell or erythrocytic cell, which is also called as erythrocytic cell, right? So now see, listen properly, again I'm saying it. See, these cryptozoites which are mainly released from this pre-erythrocytic cycle will have two options. One is it can enter into the fresh hepatic cell, right? It can enter into the fresh hepatic cell and continue the exoerythrocytic cycle, or else it can directly enter into the RBC cell, which is also called as erythrocytic cell. Okay, so now what happens once it enter into the fresh hepatic cell? Again the same process will be repeated, because this has been entered into the hepatic cell itself, right? It doesn't enter into RBC cell, it entered into hepatic cell. So same what was happened over here, the same process will be repeated, right? Hence it is called as pre-erythrocytic cycle. This is called as exoerythrocytic cycle. And what is the time period for this? Same, 8 days, right? And now here the same trophozoite will get transformed into schizont, and this undergo the process of mitotic division, right? And then it starts releasing of macrometacryptozoites and micrometacryptozoides. Okay, macrometacryptozoites and micrometacryptozoids. And these macrometacryptozoides, what they will do, they will again enter into the hepatic cell and will continue the exoerythrocytic cycle only. Now this micrometacryptozoids will have an option to enter into this RBC cell to get infected, right? So now this macrometacryptozoids will enter into the hepatic cell and continue this exoerythrocytic cycle only. Now this micrometacryptozoids will enter into the RBC cell and starts infecting it. Fine. So now this is called as pre-erythrocytic cycle. This is called as exoerythrocytic cycle. And what is the time duration for this? 8 days. What is for this? 8 days. Okay. So now once it entered into the RBC cell, what will happen? Here the actual story begins. Listen properly, please. Okay. Now this micrometacryptozoid entered into the RBC cell, right? And now this will get transformed into trophozoite, right? It will get transformed into trophozoite. And now this trophozoite will get matured in such a way that the vacuole will be developed inside the cytoplasm of the trophozoite, right? So this is the vacuole. The inside one is called as vacuole, and the extra space is called as cytoplasm. Okay, so here what happens is that this vacuole and this cytoplasm will be pushed towards peripheral region of nucleus, right? So this is not present in the center, right? This vacuole is not present in the center region. It is pushed towards upside, towards peripheral region. Both the gap, which is called as cytoplasm, and the vacuole, both of them are pushed towards the cytoplasmic region, peripheral region, in such a way that it meets towards the nucleus, right? This is a nucleus, right? And now this is a signet ring stage because it looks like a finger ring, right? Hence this stage is called as signet ring stage. And basically this trophozoite is that's what I have said you here, spherical shape. Okay, and now what will happen here? See, now in the next stage what will happen, this vacuole will be this vacuole will be off, it will be removed out. Okay, this vacuole will be absent in the next stage, and that next stage is called amoeboid stage, right? So this signet ring stage will get transformed into amoeboid stage, right? And now here in this plasmodium what will happen? The pseudopodia will be developed, right? This vacuole will be it will be degenerated, and then pseudopodia will be developed, and this pseudopodia mainly helps for locomotion, right? It helps for locomotion, and it also help in grasping the food material in order to consume it. Okay, and now listen properly. So this is a nucleus, and this is the amoeboid stage, right? And now in this amoeboid stage, as it contains pseudopodia, then what will happen? Then we know that this is the red blood cell, and in this red blood cell there is a presence of pigment called as hemoglobin. We all of us know that because we since childhood we are learning it, right? So inside the red blood cells we have hemoglobin, and now this, with the help of pseudopodia, this amoeboid stage plasmodium will consume that hemoglobin which is present in red blood cell, right? And now what will happen? Let's see. So hemoglobin has been consumed by that amoeboid stage of plasmodium, and here only in this hemoglobin the globin part will be digested by that plasmodium, whereas heme will not be digested, right? Hemoglobin will be consumed by this amoeboid stage of plasmodium, right? And that hemoglobin which has been consumed, in that hemoglobin only the globin part will be completely digested, but as the heme part will not be digested, in such a way that heme will get converted into, or else will get transformed into, insoluble crystalline called as hemozoin, right? Remember, hemozoin. And this hemozoin, it is also called as malaria pigment. It is also called as malaria pigment. Okay, now you have understood what is meant by this hemozoin. Okay, so now as it is depending upon that hemoglobin, and it is consuming that hemoglobin, right, that globin part, in such a way that it will develop its size, and that developed plasmodium is called as scisont. It's called as scisont. Okay, now that hemozoin, I have said you right, hemozoin, due to the presence of hemozoin, there is a formation of Schussner granules which are mainly present on the surface of this plasmodium, right? Now it will get transformed into the next stage in such a way that the merozoites will be developed. The merozoites will be developed, and now these merozoites will the number of that merozoites inside the cytoplasm of this plasmodium will be increased in such a way that cell wall of that plasmodium will be ruptured in such a way that the merozoites which are mainly present inside the cytoplasm of this plasmodium will be released out, right? Due to the rupturing of that cell, the plasmodium will be released out. This merozoites will be released out, and along with these merozoites the hemozoin will also be released, right, which is called as malaria pigment, that will also be released out. So how many merozoites will be present inside the cytoplasm? 12 to 24. 12 to 24 merozoites will be released once it is ruptured. Okay, so now these merozoites, right, it will have two options, two choices, where they can develop into gametocytes, or else they can again enter into the fresh RBC cell and continue this cycle. continue the cycle, and this cycle is called as erythrocytic cycle. Why it is called as erythrocytic cycle? Because it starts infecting the red blood cells. So what is the other name of red blood cells? Erythrocytic cells. Hence the cycle is called as erythrocytic cycle. And what is the time duration of this erythrocytic cycle? 48 hours. Okay, so now this is the erythrocytic cycle. Till now what has happened is nothing but the erythrocytic cycle. Okay, so now 12 to 24 merozoites has been released out, and if it attacks the RBC cell, then the process will be continued, then the cycle will be continued. And if it doesn't enter into the RBC cell, then it will get transformed into male gametocyte or else female gametocyte. And now this male gametocyte is also called as smaller microgametocyte, whereas this female gametocyte is also called as larger macrogametocyte, because this female gametocyte will be large in size, whereas this male gametocyte will be small in size. Okay, so listen properly again. We know that if mosquito will bite humans, then it sucks the blood and it also releases the material which is present inside the gut of the mosquito into the humans. So once if that mosquito will suck the blood, then what will happen? Then we know that the blood consists of this male gametocyte and female gametocyte, right? Because the cycle has been happened, so male gametocyte and female gametocytes are present. And once that mosquito will suck the blood from the humans, then this male gametocyte and female gametocyte will enter into the mosquito, right? Hope you are cleared. And once this male gametocyte and female gametocyte will enter into the mosquito, then Plasmodium vivax in mosquito phase will occur, right? So till now what was happened is nothing but the Plasmodium vivax in man phase. So once this male gametocyte and female gametocyte will enter into the mosquito, then Plasmodium vivax in mosquito phase begins, right? So that Plasmodium vivax in mosquito phase will be explained in the next coming video. Okay, so now this erythrocytic cycle is also called as Golgi cycle. It is also called as Golgi cycle. So why it is called as Golgi cycle? Because the description, the information and the explanation of this erythrocytic cycle only this erythrocytic only has given by Camillo Golgi, scientist Camillo Golgi. Hence this erythrocytic cycle is also called as Golgi cycle. I said you there are 6 stages in Plasmodium vivax in man phase. They are pre-erythrocytic cycle, which I have explained you, exoerythrocytic cycle, which I have explained you. And what is this pre-patent period? What is this pre-patent period? Let us see now. So the interval between the first entry of Plasmodium into the blood in the form of sporozoite and the second entry in the form of cryptozoite is called as pre-patent period. Again I am reading, clearly listen. The interval between the first entry of Plasmodium into the blood in the form of sporozoite and second entry of Plasmodium in the form of cryptozoite is called as pre-patent period. So what do you mean by this? They said you the first entry of Plasmodium into the blood in the form of sporozoite. So this is the first entry, right? Sporozoite, and this is the first entry. And second entry of Plasmodium in the form of cryptozoites. So this is the second entry, right? So this cryptozoite will enter into the red blood cells, or else this can also enter into the red blood cells. Okay, so this will be second entry. So this is the first entry and this will be your second entry. So that period, that interval period which is present between these both the entries is called as pre-patent period. So what is the duration of the pre-patent period? 8 days, because the what is the period which has occurred in this pre-erythrocytic cycle? 8 days, right? So now you got the logic. And now coming to the next one, we have discussed about erythrocytic cycle. And what do you mean by this incubation period? Let us see. So the period between the entries of Plasmodium into the blood in the form of sporozoite and the first appearance of malaria symptoms in man is called as incubation period. So what do you understand by this sentence? So they said you that the period between the entries of Plasmodium into the blood, which means the entry of Plasmodium into the blood means, so here the Plasmodium has entered into the blood, right? So this period and the malaria symptoms which was appeared in the human being. So the both periods, I mean the you know what, the interval between the both of these periods, which periods? The first period is which enters into the blood, I mean the sporocytes which enter into the blood. So this will be the first period, and the second period is nothing but where the malaria symptoms which you can see is the second period. Okay, and that interval period which is present between both of these periods is called as incubation period, right? And what is the duration of that incubation period? 10 to 14 days, right? 10 to 14 days. And now, so here the sixth stage is called as formation of gametocytes. So I already explained you about the formation of gametocytes, the male gametocyte as well as the female gametocyte, and this is about the Plasmodium vivax in man phase. And one more important thing which people have to remember is that if this male gametocyte and female gametocyte doesn't enter into the mosquito within 1 week, then this both will get degenerated, they will get expired. Okay, so this is one of the things which you have to remember. So this is about Plasmodium vivax in man phase. I have explained you each and every point of Plasmodium vivax in man phase very clearly. I hope you understood this topic very clearly, and if you catch my point clearly, then you can understand this topic, because it is very simple and easy, and it is highly asked in your NEET examinations and even for each and every medical entrance examinations and your MCET also. Okay, I hope I cleared this topic for you, and if you like my explanation, like the button, okay, and also press the bell icon for the latest updates. Thank you.